So we now have practically single-cell atlases for every human organ, every model organism, as well as the different disease states of our human body. So out of the approximately 37 trillion cells in the adult human body, we now have a pretty good categorization of their states and their stable types. Okay, so these cells are arranged in our body in tight-knit communities. So every one of us is actually a community of cells. Being multicellular organisms, we're a community of cells working together to carry out the biological functions. So this is a slice of the human colon, just as an example. This is what we call spatial proteomic data. So this is obtained by labeling proteins, and using the proteins, we can now know where the cells are and what cell types they are. So these are just examples of the spatial atlases that we can now build for our human tissues. So from this kind of data, there's many questions that we are now in the process of trying to understand. For example, what cell types are in the human body? I think we have basically answered that question already, which is pretty revolutionary, because 10 years ago, I cannot say that we know what cell types there are in the human body, but now we do. How do they form? So from an embryo that started out to form a fetus, how do the different cell types differentiate from the embryo? What roles do they play in the human body? So that is a question that we still don't know how to answer. Okay, so for many cell types, we know that, for example, neurons fire to make us think, and our white blood cells fight diseases, but we don't actually know how exactly they perform these functions and how they communicate with other cells. So this is still very much a hot topic of research. How did the cell types change between health and disease? I'm going to talk more about that. And now we're in the process of learning how to program our cells through various new technologies such as CRISPR perturbations to perform functions that they were not programmed to do by nature. Okay, so lots of very exciting developments. So out of this, this 1 hour talk, I'm just going to focus on 3 computational challenges. First, I'm going to talk about the problem of data denoising and variance decomposition. That is an old topic. This is the paper that the Frontier Award was on. I was told I'm supposed to talk about it. So the first part of my talk will be on that topic, which I think sets the foundation for the second and third topic. The second topic is a more biological question of how do we estimate transcriptional entropy, and I will tell you what that is and why we want to estimate it, and how we can use that to characterize cellular aging. And then the third question is, how do we do single-cell and spatial data integration? So I'll give more background on this as we move along. So first, data denoising and variance decomposition. Okay, so here I'm just going to focus on these 3 papers that came out a few years ago, where we figured out how to model the technical noise in single-cell data and then remove that technical noise to reveal the biological structure in these cells. So I just want to show you this graph to give you a sense of the scale of the data. So the first single-cell data set came out 2009, and that was a really big deal when they were able to sequence the whole transcriptome of 9 cells. So that data set only contained 9 cells, and that was very, very expensive. But over the years, you see this axis is on the log scale. So most single-cell experiments now can collect upwards of a few 100,000 to millions of cells per experiment. Okay, so the scale has really just exploded with the technological advance. So each of this is a different technology for sequencing single cells. Now, A typical experiment, so this is in terms of number of cells, right? Millions of cells per data set. But within each cell, how many molecules do we sample from the RNA soup that makes up that cell, right? So you can think about each cell. Within the cell membrane, there is many RNA, so DNA makes RNA, makes protein. The protein is the machine that does the work, and the RNA is the intermediate. So we sequence the RNA in order to figure out what the cell is doing. So a typical experiment samples about 1,000 to 5,000 unique RNA molecules per cell. Okay, that is the data. And then the actual RNA pool, so it's how much, so that's really downsampling, because within a cell, there's usually a couple 100,000 transcripts. So we're trying to figure out using this limited sample, using this limited sample, what is the state of that cell. So when we say data denoising, what we mean is that, as you sequence more molecules per cell, you're going to see more structure. So let's say that there are 3 cell types. Okay, your cells could come from 3 different cell types. So that would be 3 different clusters. If you were to sequence 1 million reads per cell, then you're going to see that structure very clearly, whether through a PCA or through a heat map. However, nobody can afford this. Most experiments are somewhere here where you don't really see much structure. Okay, so the data is kind of murky. So what did we do? We did this thing called Saber denoising, which basically takes a data set that looks like this and makes it look like that. Okay, so what is this? So this is a UMAP embedding, right? So if you guys heard of it, the UMAP is a way to take high-dimensional data. So your data is a matrix, a cell. Each row is a cell. Each column is a gene. And the entries of that matrix is how many copies of that gene do you see for each cell, right? So it's a row by column, cell by gene, okay? So what we're doing is we're clustering the rows, right? So we're taking each cell, which is a high-dimensional vector, and putting it onto a 2-dimensional embedding to visualize it. And then, so the original data would look like this. They are colored by their known cell type. So this is for a data set where you have expert biologists going in and annotating the cells to tell us what they are. So what you see here are B cells, monocytes, NK, natural killer cells, T cells. So if you have studied some biology, you know that these are the cells of your blood. Okay, so they're blood cells, different kinds of white blood cells. And then so as expected, for example, monocytes and somehow it senses that I'm doing this. Okay, so somehow monocytes and B cells look very different, which is good because they are very different cell types. But this cluster is all mixed up, and these are the T cells. Okay, so I haven't told you how we did it yet. I'm just showing you that after you do denoising, you can make these T cells separate out into their separate clusters, which is what we want, because we want to be able to differentiate between them, right? And that is very important because these T cells are sort of like the soldiers in our body to go and kill the germs, right? They go and kill different bacteria or different viruses that invade our body. And these T cells can be divided into subtypes with very specialized function. For example, there are memory T cells, regulatory T cells, there are cytotoxic T cells, which are actually the killer T cells. So it's very important in diseases to be able to tell them apart. In fact, there's many different subtypes of cytotoxic T cells and such. So being able to differentiate that structure in that population is important, and that is what denoising is trying to do. Okay. So this work was like, it's old. It's 7 years ago, okay? And, but it's still relevant because there has been a lot of, what I'm listing here are basically benchmarking papers and review papers in the field that try to take all the, there's many other denoising methods, which I don't have time to talk about. They try to benchmark them. And basically, if you look at all of these papers, they have their recommended method, and SAVOR was one of their recommended methods. And the latest paper was this one, which is published in Cell of last year, which does a sort of differential expression analysis for single cell data. And they have also benchmarked SAVOR 7 years since its publication, showing that it's one of the best methods in the field. So I'm not going to show you our own benchmarking. There's a lot of third-party benchmarks which you can rely on. But I will just show you one, which is the first one here. And they made the point that this kind of denoising, or sometimes called imputation for single cell data, because they call it imputation because you lose a lot of genes because of the sparse sampling. They say that this kind of imputation is kind of dangerous because if you don't do it well, you can generate a lot of false positive results, meaning you generate spurious structure in the data. So, so they say, don't do it unless you know what you're doing. But out of all the existing methods, SAVOR is the one which can recover the underlying structure without the false positives. And then here's another third-party benchmark that was published in the Nature Methods that show that if you use SAVOR, you can actually get the best. What did it do? It's correlation between replicate experiments. So it's been validated by other people in the field. So how does it actually work? The idea behind SAVOR is really simple. Okay, what it does is a variance decomposition of single cell data. Okay, so here's the data, as I told you, is a matrix. Each row is a cell, each column is a gene. There's about 20,000 genes in our genome, and each experiment is about, at the time it was published, each experiment was about a few thousand cells. Now it's millions of cells. Okay, so these cells are high-dimensional vectors because there's many genes, 20,000 genes, but they are actually live close to a low-dimensional manifold. So it's about manifold. It's all about manifold fitting. So these experiments. So when you read single-cell papers, I'm not sure if you guys, any of you, are familiar with this data, right? So what you oftentimes do is to take this matrix and do a PCA into like 50-dimensional space. And with that, in that PCA space, you would do a graph-based smoothing, a graph-based manifold fitting, and then visualize that on a UMAP. And then many of the things that we do with single-cell data downstream, such as clustering, cell labeling, trajectory, or pseudotime analysis, are all based on that fitted manifold in low-dimensional space. Okay, so what SAVOR does is it models the data through a hierarchical model. So YGC is your observed data. We assume that it is Poisson with mean, that is the library size of the cell. Library size meaning it's just cell-specific size factor that can be estimated, multiplied by the true expression XGC. Okay? And that true expression we assume to be gamma distributed with mean lambda GC, which is this learned manifold, and with a gene-specific dispersion, phi G. Okay? And that's it. That's the model. Okay, it's very simple. But each component of this model has been validated by experiments. Okay, so we had a paper in PNAS that shows that the technical error is indeed Poisson. Okay, with this library size. So when I say indeed, it's well approximated by a Poisson distribution. And then we had experiments that help us convince at least the reader that this that basically the true expression can be modeled this way with a gene-specific, what we call intrinsic entropy term. Okay, so based on this, what we can, I'm sort of just showing you, sweeping a lot of technical details under the rug. What you can do here now is do an adaptive shrinkage to the manifold. What that means is you can estimate XGC, that's the quantity of interest, right? We want to know what is the true expression of the gene in that cell. So we call that X hat GC. X hat GC is a weighted average of your true expression, your observed expression divided by the library size plus the manifold fit. Okay, so it's sort of, you have the manifold fit and your estimated expression value is somewhere in between the observed expression and the manifold fit. And these weights, WCG, which I'm not showing you the formula of, these weights can be computed from the data. Okay, so the weight depends on the LC, the cell-specific library size, as well as phi G, which is the gene-specific dispersion. So it depends on both technical noise as well as biological predictability. Okay, so I don't, that's all I'm going to show you about SABRE, okay, because I should tell you a little bit about this work. I'm going to go on later to this is unpublished work on transcriptional entropy, right? But what I want you to take away from the SABRE work is that we can decompose the data into three components. Extrinsic variation, which is that manifold, plus intrinsic cellular noise, as modeled by that phi G term, plus technical noise. Okay, so our paper was the first to give this hierarchical decomposition of single cell data. And then with this decomposition, the SABRE denoising is just really simple. It's basically adaptive shrinkage of each cell to that manifold fit, and that adaptive shrinkage accommodates both technical noise, which you can estimate, as well as biological uncertainty, which you can also estimate on a per gene basis. Okay? So next, let's talk about transcriptional entropy and cellular aging. Okay, so it's been 7 years since we sort of finished that work and submitted the paper. So we didn't know that it would take this turn. So this was quite a serendipitous discovery. Okay, I'll tell you where this comes from. Okay, so in order to appreciate this work, you have to look at what we mean by cellular aging. Okay, so organisms age, right? Each of us, you guys are all young, most of you are students, right? So, organisms age, right? We're well aware of that. We know how each of us has a biological age. But what do we mean by cellular age, right? Is that even a meaningful scientific concept, right? Because many of our cells are very short-lived. Our red blood cells, there's a high turnover, right? So most of our cells don't actually age with us, right? Or do they, right? So we don't actually know how to quantify cellular aging, okay? We know, there has been work that try to quantify tissue aging, right? So with aging, as each of us age, our tissues change, right? So there has been plenty of bulk tissue studies that try to measure gene expression changes in our tissues as we age. And we know that there's drastic changes. If you take the blood of an old person and the blood of a young person and compare them, they have very different gene expression signatures, okay? So that is well characterized. However, so for example, there's been studies in the mouse where they, any humans where they have characterized in detail that certain cell types increase with age and other cell types are depleted with age. For example, there's age-associated B cells, there's activated regulatory T cells, our regulatory T cells increase. You have less of certain macrophages. So this is well characterized, right? So what we know is that a lot of our aging, the aging signatures in our tissues are driven by changes in cell type composition, not single cells aging, not specific cells changing, okay? So it's basically a population shift rather than individual cells age, okay? The question then is, as we age, how does that change our individual cells? Okay, so that is what this new consortium called SENET is aiming to do. So this is a big consortium involving many institutions in the United States. It's an NIH-funded consortium that is trying to tease apart what happens to individual cell types when they age. Okay, and we do have some clues on what's going on. For example, as our cells divide, so some of our cells, like our stem cells, so every one of our tissues have their tissue resident stem cells. These cells are long-lived. They live almost as long as us. Okay, and they are sort of our protected tissue niches, replenishing they help with our tissue regeneration. For example, our skin, right? It keeps regenerating because there are stem cells. So these cells keep dividing, and every time they divide, the DNA could be damaged. There could be mutations, right? So there is DNA damage in our cells as they age. Okay, there are epigenetic changes. So for example, our cells have this specific chromatin formations that become like damaged as time goes on. There's telomere erosion. There are large proteins that should be cleared, don't get cleared, so there's aggregation of large proteins. There's chronic inflammation. So we do know that at the cell level, bad things happen to them as individuals age. However, how do we measure that, right? So there has been quite a number of papers that try to develop aging clocks for our cells. So what they do is based on expert knowledge, they curate a bunch of genes that they think are related to our aging, and then they basically put them into a black box machine learning algorithm where they feed it like labeled data sets. So they take, for example, blood from old people and young people, and old mice and young mice, and then they are labeled by the organism age, and then they would see which genes predict that organism age. So there's a Y, which is the organism age, and then they try to find genes that predict them. So they get a bunch of cell senescence scores based on these gene lists, but they don't really work. So for example, here is this is the age of the mouse, 3 month to 24 month. 24-month mice are like 70-year-old people. Okay, so they are old people. So you have this gene list that is selected and optimized for bone marrow, and then you see that it predicts the chronological age of hematopoietic stem cells. However, if you take this gene list to predict the aging of bladder, then if you train it on the bladder, then it doesn't predict the stem cells. So what this is showing is that it really these machine learning based aging measures really depend on the training data set, because this aging phenomenon is very cell type specific. So different cell types break down differently, and even for a given cell type, what we found is that different organisms, there's different process of breakdown. Because, it's just like when people age, right? There are many things are unpredictable, right? Depends on environmental factors, depends on random assaults on the human body. So aging trends are not easy to predict in organism level. It is even harder to predict on the cellular level. Okay, so despite the SENET project, that the SENET consortium has been funded for 3 years now, there's still a lack of good aging markers or cellular aging markers. So because of this, what we brought to this picture is that we had this revelation that aging at the cellular level is just a continuous process of damage accumulation. bad things happen to the cell, and oftentimes they're random. There's really no fixed trend. And what we want to measure is just the gradual deterioration or the increase in entropy at the RNA level of the cell. So our goal is to use single-cell RNA-seq data to get a cellular aging clock based on some measure of entropy. So now let's get back to that decomposition of variation. Remember that our data comes to us as a matrix of cell by gene matrix. And we assume that the cells basically lie close to that low dimensional manifold that we fit to the cells. So you can fit this manifold using a convolutional neuron, like autoencoder, a variation autoencoder, or some simpler method, right? So there's many ways of manifold fitting, right? So you can fit your manifold, and then you can assume that the cells lie close to that manifold, right? The question here is, you could be in a high entropy scenario where the cells are very far from the manifold, or you could be in a low entropy scenario where the cells are close to the manifold. And that's actually what separates out SAVOR from the other denoising methods, because other denoising methods just place your cells on the manifold. It does not assume there's a biological dispersion. Okay, so in SAVOR, we say there's a technical noise, there's an underlying manifold, and there's this stuff in between, which is the biological entropy. Okay, and with that, we can decide whether we're in which scenario we are in. Are we in the scenario where the cells are far from the underlying embedding or close to the underlying embedding? So we thought, maybe that is the key for us to understand cellular aging, because young cells should behave in a very coordinated way, in a low entropy way, whereas old cells should behave in a high entropy, unpredictable, idiosyncratic way because of all the natural assaults it has sustained through its lifetime. So, given this insight, the mathematics is quite simple. We develop two measures of entropy, one at the cell level and one at the gene level. So you give me a cell, I can tell you how messed up it is, and you can give me a gene, I can tell you how much entropy there is in that gene for that group of cells. Basically, it's based on distance from that estimated manifold. So we went and said, okay, we have this measure. What is it? How do we convince biologists that it is meaningful, right? Because it's a mathematical quantity. They don't understand, when you say manifold and things like that, it's very hard to convince biologists that this is something that they should care about. So we went back into the literature, into the classic, are there classic definitions of intrinsic cellular noise? Okay, so we found this experiment conducted by these four authors, Michael Elowitz, Levine, C.J. and Wayne. This is a very famous study where they studied bacteria. So what you're seeing here are E. coli labeled with two different dyes, the red dye and the green dye. And these two dyes are given the same sort of reporter sequence, so they should be transcribed together. So they had bacteria with these two dyes, and then they imaged them. So they shot videos of these bacteria, and you should, I don't have the videos, and then you see each bacteria is a color. If it is green, that means it's only expressing this one. If it is red, it is only expressing that one. And if it's yellow, that means it is expressing both. So if there is no noise in the system, if this gene is very coordinated, you should be seeing mostly yellow, because both of them should be expressed at the same time. But what they found was that there was a big difference between bacteria. So what they basically developed is a measure. Sorry, I'm just gonna move this lower. So what they developed is a measure of the intrinsic noise based on the difference between these two dyes. Okay, so the red minus green, and that is sort of the measure of the difference between the sort of the stochasticity in the system. So we asked this question, is our measure of intrinsic noise in the single-cell RNA-seq data the same as what these classic experiments, which are based on imaging, trying to measure? So we went to a system where there is an F1 cross of mice. So F1 cross are where you have two purebred mice and go to the first generation cross. And this first generation cross basically has many heterozygous positions where these two breeds of mice differ. So it's heterozygous, so you have two different alleles at these heterozygous positions. Then what you can do is actually mimic through this system that bacteria experiment where you have two alleles of the same gene. And then so we can conduct single-cell RNA sequencing on this F1 cross of mice and then take the difference between these two alleles, and that is our sort of single-cell mimicking of that classic experiment. And on this, we can compare our manifold-based estimate of gene level noise versus the allelic discordance, which is basically that classic definition, and we found that there is a high concordance, sort of as expected. So that means that this manifold-based measure is a measure, another way to get at the intrinsic cellular noise that was defined in these classic experiments. Okay, so that sort of is nice because we can connect it to these classic definitions, and then we can ask that what does this intrinsic cellular noise tell us? So we looked at an experiment where cells are exposed to ionizing radiation, which is supposed to inflict heavy damage to the cells. So you see that when the radiation dosage increases, our gene level noise increases. So that basically means that when there's no radiation dosage, we're in a low intrinsic noise scenario. When there's a high radiation dosage, we're in a high intrinsic noise scenario. What that means is that intrinsic cellular noise reflects the amount of damage a cell has sustained. Okay, so it's a meaningful measure of damage. And then finally, let's look at aging. So this is we looked at this data from this consortium. It's called the Aging Mouse Consortium, Tablet Mirrors Cenis. And what they do is they take mice from different age strata and then did single-cell sequencing of all of their organs. And then so we specifically looked at the bone marrow, and we specifically looked at the hematopoietic precursor cells in the bone marrow. Why did we do that? Because these are the stem cells that help you regenerate your blood. So stem cells used in your blood is you don't look like red blood cells because those are so short-lived, right? White blood cells are short-lived too. You want to look at the stem cells, okay? So these live in the bone marrow. And then so we extracted the stem cells, and then we plotted them on a UMAP and compared the different age groups from 1 month to 30 months mice. There's no difference. You can't really find a cluster that is age specific, as expected, because if it were so easy, then people would have already gotten the markers, right? So, but then when you computed the gene level dispersion, which is what the aging clock that we developed, you see that indeed it increases monotonically with age. Okay, so that was a validation that this dispersion, this entropy-based clock is a measurement of cellular aging. Okay, so there's low intrinsic noise for young mice and high intrinsic noise for old mice. Okay, and what is even more interesting is that if you compare between cell types, there's the stem cells, the hematopoietic stem cells. And then here are the monocytes, which are the well-differentiated cell types. So as you go down this axis, you're going from stem to differentiated. These are the multipotent cell, the potency increases as you go there. So you see that stem cells age with the organism, whereas differentiated cells do not. So that was, a revelation. Right? So not all cells age in the same way. Okay, so we compared it to the black box machine learning based methods. I want to go quickly through this because I've been going too slow. So, we found that in different tissue niches, the bone marrow, the lung, the limb, the adipose, the stem cells always increase with age, whereas if you use, like, for example, a Mayo score is one of the best machine learning based method right now. It has a less obvious trend. Okay, so if you look at the scatter plot of the Mayo score with the intrinsic noise, the entropy, you see that it matches for the bone marrow, but not for the other tissues. That is because the Mayo score was trained on the bone marrow. So it does well for the bone marrow, but it doesn't carry across tissues. Okay, so let's look at the bone marrow. Here's the data for the bone marrow. Here's the Samayo enrichment score on the x-axis, the intrinsic noise on the y-axis, that's our metric. And you see that they disagree for these cells. These are the cells that we say are very old, but the Samayo score didn't fish them out, right? So if you look at these cells and plot out their age strata, you see they mostly come from old mice. So these were missed by Mayo, but found through intrinsic noise. Okay, let's look at another example, rat kidney. So we had a collaborator in Penn collecting kidney cells from 4 age strata of aging rats. Okay, the reason why we look at kidney, because kidney is a blood filter. Okay, so our blood carries a lot of toxins. So these kidney cells sustain a lot of stress. Okay, and they perform what's called single cell multiome on these cells. So looking at both the RNA and chromatin, we computed RNA entropy, and we found that in different segments of the kidney, segment 1, segment 2, so the kidney is a blood filter. So there's a tubes, right? So this is a nephron in the kidney, and then our blood flows through this and to get filtered. And there are 3 segments of the proximal tubule, which is this tube here, segment 1, segment 2, segment 3. And we found that in segment 3 and segment 2, there is an increase in age of these cells as the kidney ages. And our collaborator says, well, you know what? That is exactly what we suspect. Because during chronic and acute kidney injury, the segment 3 is the most affected, while segment 1 is usually the most protected. So oftentimes in kidney injury, it is the segment 3 cells that become damaged. So what we have been able to do was to use this computational measure to extract out from the tissue the cell types that are affected by disease, right? So that's a validation of this measure. Okay, so I'm gonna skip over this, but I'm just a summary of what we were able to find. Okay, so we started with a statistical hierarchical model that gave a variance decomposition of single-cell RNA-seq data. In this model, it decomposed the variation in the single-cell data into 3 components. The extrinsic variation, we call that the extrinsic variation, it's basically the manifold fit, the intrinsic cellular noise, and the technical noise. And then we formed an estimate of the intrinsic cellular noise, and we showed that it is consistent with classic definitions of intrinsic noise based on the dual reporter bacteria assay. And then we show that it reflects the amount of damage a cell has sustained, and that it gives us a first principle based unsupervised aging clock at the single cell resolution. Okay, so that's sort of like, we were very happy that, this statistical measure that came out of our attempts to denoise the data, in the end gave us a very biologically meaningful quantity to be used to stratify cells based on their amount of damage. Okay, so that's the second part. So with the remaining time, I want to just tell you another work that is, come from this pervasive computational task that comes, that you see in every single cell workflow. It's called integration. This I've been telling you about variance decomposition. When we do, I just want to show you this work because this is a different variance decomposition that turned out to be meaningful. So, okay, let's go into it. So, any of you actually work with single cell data? No one here work with single cell data. Okay, so that's good because I'm going to show you go back to the background of what is cell integration. Okay, so I'm going to show you two examples of single cell studies to sort of illustrate what we mean by integration. Okay, so in this first study, we're looking at human colon. Okay, and the goal of this study is to just form an atlas of the human colon. What that means is they take fresh frozen human colon and they did 3 experiments on it. This codex imaging, single nuclear RNA-seq, and single nuclear ATAC-seq. Codex imaging is a spatial experiment. So I told you this is talk is single cell and spatial data, right? So this is a spatial data. The spatial data is you know exactly where it is in the tissue. You don't lose the spatial coordinates. In single cell data, you pull apart the cells and you dissociate the cells, so you see each individual cell, but you don't know how they are arranged in the tissue. Okay, in the spatial data, you know what its neighbors are. So that's what the codex imaging gives you. And then you have the single nuclear RNA-seq and single nuclear ATAC-seq. The RNA-seq is what we've been talking about. It's cell by gene, the RNA expression. ATAC-seq tells you about the DNA conformation. So it's not about what RNA is expressed. It tells you which genes are exposed and ready for expression. Okay, it tells you, so it gives you a more like a lower level layer of information in the cell. Okay, so they collected all 3 data types because they want to have a full picture of what's going on in the colon, right, at the single cell resolution. But this is not ideal because given a slice of tissue, you either do this or do this or do this. You can't measure RNA and ATAC on the same cell. Well, now you can, but they didn't do it at that time. And then you cannot do all of this while keeping its spatial coordinates untouched. So what we would like, ideally, is to measure all the modalities on the same cell mapped in space, right? But each experiment gives you something. There's no experiment that gives you everything. So in this case, what we want to do by integration is to integrate all 3 data sets so it feels like we're seeing the same, every modality on every cell. Okay, so that's one example. I just want to show you another example of cell integration to show you how varied the integration problems could be. So this is a case where we're looking at kidney when we want to study chronic kidney disease. This is with a collaborator also at UPenn who is a kidney expert. And what she did was she collected kidney samples from healthy individuals. So these are mostly donor tissue. Okay, healthy individuals as well as individuals with chronic kidney disease. And from each sample, she either did single cell RNA-seq or single nuclei RNA-seq. So the difference between here is cell and nuclei. Okay, cell is the whole cell, nuclei is just a nuclei. The reason why they did different things is because it depends on whether the sample came to you as fresh or frozen. Okay, sometimes they can't give you the fresh sample because it's collected at a hospital where they have to process it right away. So this is not something you can control, okay? So if it's fresh, they can do single cell RNA-seq. If it's frozen, then they cannot pull apart the cells because it's all frozen. So they can only do single nuclear RNA-seq, okay? So each sample, some samples have both. And then, so what they would like to do is to compare between the disease and normal to see what is different. But then the problem is that once you take all the cells, there's 200,000 cells that came out of the study, so 200,000 rows in your matrix by gene. And if you just plot them out, you see that yes, the control and the disease do differ. That's what you want to find. But If you color it, so don't blink, if you color it, this is control versus disease. But if you color it by protocol, single cell versus single nuclei, you see that that's an even better separator. Okay, so most of this is probably just technical, nothing that is really biological. And in fact, if you just, okay, now if you look at within protocol, if you color it by sample ID, you see protocol, sample ID, you see that it's not just single cell versus single nuclei. Even within the same protocol, just different samples, depending on how long it rested on dry ice, or who's the technician that did it, or what instrument you processed on. All of this matters to make the samples look different. And that's not good, right? Because that is all technical. It's all confounding factors. So what we would like to do here when we do single modality integration is basically batch correction. We want to remove all that unwanted technical effects to get the actual biological structure. And I just want to show you an example of what happens after you do data integration. Instead of seeing a mess like this, you get very clean clusters popping out, and these clusters actually represent different cell types. Okay, they should represent the different epithelial cells or immune cells that are in the kidney. Okay, so with the remaining time, I think there's only a few minutes. I just want to, I don't want to go too much into the details. I just want to say that there are different setups, different goals, right? I showed you just now the kidney atlas example, where our goal is to identify disease versus healthy differences, versus the human colon atlas example, where you have the exact same tissue, but processed in 3 different layers of information, right? Protein, RNA, and ATAC. So the goals are different. In the first kidney atlas example, you're looking at actually different biological contexts, disease versus healthy. The same modality, you're getting RNA from all of them, so it's the same modality. And our goal is basically to compare these tens and hundreds of thousands of samples in order to figure out what are the biological differences, okay? Whereas in the human colon atlas example, you only have one biological context, which is this donor tissue that is lying in front of you. And so same context, but different modalities. And when you integrate, what you want to do is to align the cells between the different modalities. So each cell, you either see RNA or chromatin or protein, and you want to be able to align the cells between modalities in order to form a more unified representation of this tissue. Okay, so in both cases, what you want to do is align the rows of these matrices, but the goal and the context is very different. Okay, so these are like 2 of the many different flavors of integration that you run into in single cell and spatial omics. Okay, so we worked on this we worked on both problems, and in the first case is a single modality, so like think kidney atlas, same modality comparing biologically different samples. In that context, what happens is that oftentimes people are too aggressive in trying to integrate the data. So when they integrate data across these hundreds of samples, they end up just erasing the biological signal. Okay, so we developed a method to recover that biological signal by harnessing different aspects of this experimental design. And that's called CellAnova. In the second case, what you have is different modalities, RNA, ATAC, protein, whatever, different modalities, but they're all measured on the same biological sample. And in this case, what we did was we developed a method that can sort of integrate across weakly linked modalities. So in the interest of time, I'm just gonna take 2 minutes and go really quickly over what the key idea behind CellAnova is. Okay, so this is this example. CellAnova is about these kind of disease atlases where you have different biological context, like, healthy disease, treated, untreated, okay. And so I just want to tell you, because you guys are mathematicians, right? So you want to know about the motivation. I'm not going to go over the technical details of our solution. I'm just going to tell you that when the motivation for this problem, when we got this data from our collaborator, she already tried all the integration methods out there. And the two that are the most popular, among the most popular, is called Seurat and Harmony. Seurat has like 12K citations despite being only out for a few years. And Harmony also has about 5,000 citations. So you see that these are very popular methods. And she tried Harmony's integration, and it did give her this beautiful UMAP with clusters that should represent the cell types. And then she wanted to focus on within cell type differences between healthy and disease, right? But she found none. There's no cell type specific disease signatures found with Harmony integration. Okay, so yeah, so that is also true with Seurat. So they all erase all of these differences. So I'm going to skip over this because it's too much detail. So what we felt was that these methods don't make use of the experimental design, because what she did was actually she went out of the way to get these control samples. It's very hard to get healthy samples because who that's healthy would give you the kidney sample, right? It's very hard. So she actually, this is donor tissue from people who donated tissue when they died. So she scored, to get this donor tissue, to get 17 donor tissue, right? And she asked us, like, how can we use this in the integration? Because these integration methods basically just squash the data. They try to merge the data together. It was blind to the experimental design, where she had a very nice case control design. So basically what we did was we used the control pool samples to estimate the batch latent space. So it is a latent space in the high dimensional gene expression space that capture the technical unwanted variation, and we rotated the data to so that we only see the perpendicular of that, the null of that space. And that was it. So I'm gonna skip ahead, and because I think I have 5 minutes. Yeah, so let me skip, okay. So I just wanna show you that without cell-ANOVA signal recovery, they weren't able to get any significant pathways in any of these key cell types in the kidney. But with cell-ANOVA recovery, we were able to find many pathways that are changed with the disease. And to convince ourselves and our collaborator that this is real, we went to an independent study. So there were two sort of groups that were doing kidney disease atlases, and they were using completely different samples, completely different sort of labs. And this was another independent study, so we sort of applied our method to both, and we found the same pathways. So the yellow highlights are the same are shared pathways, and even more so, we took one of the top pathways, this TNF alpha signaling, and then we validated on a different technology, which is a spatial technology, which does not require cell dissociation, because you just slice the tissue and you don't have to pull apart the cells. That is what introduces most of the batch effect. And we found that on the spatial technology, we are also able to identify this pathway as being altered in the disease. So, so in single cell, so that's a third topic, single cell and spatial data integration. I told you that there are two flavors of cell integration. One is between different modalities that share the same biological context, and one is within the same modality, different biological context. With this time, I'm only able to tell you about the second one, and there, what we have developed is a method that is very quick. It is a linear method that only adds minutes to the standard protocols, and it is able to reduce the data distortion and recover erased signals. And I just want to thank all the students that did this, contributed their ideas and efforts to this work. Saver was done by Mo and Jingshu. They've already left the lab. The transcriptional entropy work is done by Eddie Yang, right there. And then Ruonan Zhang is a student who did the cell integration. And I want to thank my collaborators as well who contributed data to this work. Thank you.
Right, last talk. Any questions? So in your bird observation, that you effectively write to the next binomial as Poisson as a gamma mixture of Poisson.
Yes.
So somehow this component has valid biology computation.
Yes. Yeah. The negative binomial model is used a lot in genomics, right, to model RNA-seq data. What we found, first of all, is we were able to sort of use a lot of data to validate that the Poisson in the part of the negative binomial is indeed a good measure for the technical noise. And then, because of that, we can now interpret the remaining variation, putting that as either in extrinsic variation or intrinsic variation. So after we removed the technical noise, we gave our own interpretation to the underlying parts. Yeah. The F1, you mean the mouse, right? The F1 cross. Yeah. Yes. The imprint, yeah. Yeah. Sorry. We're almost there. Yeah, so what happens when there's true allele-specific differences between the two alleles, right? So then it will be a mean difference, not a variance difference, right? So that's what you're asking, right? So, yeah. So there would be genes that where there is allelic discordance that is not due to intrinsic noise, but due to differences between the two, right? So so first of all, that is rare. We are assuming that is rare. Yeah. Between these two species, the Cas and the C57. So when you say 200 imprinted genes, what do you mean? In the mouse genome. Yeah, so we removed those on the sex chromosomes, right? The sex chromosome is imprinted, so we removed those. Even the even the autosome has imprinted genes, so those are rare. And in fact, you could model that. So we actually removed the imprinting effect in our estimate, because you can estimate that mean and then remove that mean. So you can actually observe an imprinting effect versus a noise difference. So we did remove that. But I'm saying even if we don't remove it, it's a few 200. We don't think it's a common phenomenon.
Do you have sense that we how much of the back is done by the GC bias, especially considering the last taxon?
Yeah.
If you follow the GC bias, does it help you to remove majority?
Yeah, so what you mean is that, the GC content bias for people in this room. So, the DNA, these are sequencing experiments, right? So cDNA, the DNA is ATCG, right? The CNG has 3 hydrogen bonds, A and T has 2 hydrogen bonds. So that makes it there to be a fundamental difference in the chemical properties of AT-rich or CG-rich oligonucleotides. So a lot of the biases in sequencing experiments manifest as come from this difference in the amount of CG nucleotides in the gene region that you're sequencing. So in DNA sequencing, in the whole genome DNA sequencing, for example, what was found was that GC content actually can predict a lot of the batch effects. Frankly, we did not look at that. We did not try to model that. We didn't do it. I wouldn't be surprised if some of the batch effects can be explained by GC content of the last exon of these genes that were, because this is basically a 5', either 5' or 3' sequencing. So, however, others have looked at it, and for example, in both bulk and single cell, there were efforts to try to estimate those biases, right? And for example, Wing Wang had this paper where they did try to use the sequence content to try to estimate the biases, and those models had limited effectiveness. You know, they did not really, you know, they were. I didn't think they were not really good at removing the batch effect. So in the end, instead of using the sequence information, people have gravitated towards these latent factor models, which is, you know, underlying, you know, even in the bulk setting, right? So that is why we didn't even bother to look at the GC content bias in this setting.
So in the bulk,
Bulk RNA-seq. Did you compare it with the latent factor models that came out? And they were better, or it's giving you most of it? Yeah. So I think that might, you know, maybe we'll look at that next.
I have very stupid questions, and I'm not familiar with this field, but I'm quite interested in the case. It seems like maybe the results highly depends on the quality of the data. And of course, in biology, it's different from computer science or math. I believe like sometimes you really need to, you know, spend time or money or something to collect your data. I wonder for different cells or different problems, and what kinds of data are easy to collect, but what kinds of data are not so easy?
Collect?
Yeah, to collect.
Yeah. That's a good question. I mean, it's
What kinds of data can be collected almost for free? But what kinds of data is very
not
expensive for a long time?
this data is very expensive. But that's a very good question, because this kind of genomic experiments with sequencing is always very expensive. So a running theme in this field is how do you use cheap data, like data that you can collect for maybe a few dollars per sample, and from that to impute or to sort of harness, like integrate with this more expensive data, so that you can transfer the information you learn from the expensive data and onto this kind of cheaper data. So just for example, single cell data is very expensive, right? Because for tissue, if you want to do single cell sequencing, you're sequencing thousands of cells, tens of thousands of cells, millions of cells, and then, you know, but bulk sequencing is relatively more cheaper, right? Bulk sequencing is just it gives you a vector instead of a matrix, right? And then there's a lot of work in saying, okay, now we have a single cell atlas, which tells us about all the different cell types in a tissue. How do we use that to deconvolve a bulk tissue, right? So instead of just sequencing single cells, you just sequence bulk tissue and then computationally impute the cell types in that tissue, right? So that's a way to use the cheaper data to get the kind of like cell type specific inference that you wouldn't have gotten, without a single cell atlas. And then, for example, spatial data. I showed you the spatial, beautiful spatial proteomics data, right? That is very expensive to collect. But then that is needed for discovery, because we use that to build an atlas to figure out what is in our tissue. However, H&E data, like the kind of histology data, right? Histology data. If you go in to get, sometimes the doctor will show you, maybe when they, like, okay, a cancer patient, when they get a tumor removed, you do an H&E, right? The kind of staining. That is very cheap to collect. That's only a few dollars per sample, right? So now there's a lot of effort from an H&E image to be able to impute the spatial genomic kind of experiments. Right. So maybe that helps you understand. Yeah. So, and another thing about this data is that, okay, I'm telling people in this room, I always tell this to our statistics students, and don't bother with bad data. Okay, don't waste your time, because we're in an age where there's a lot of data, right? We can always do better experiments. As we have to be very picky about the kinds of data we work with. What we want is to get scientific discoveries, right? So don't bother with bad data, find good data. It's worth it to find good data and good collaborators to give you good data, instead of trying to work with bad data that will be basically discarded in a few years.
Thank you. Any other questions? Yeah. If not, let's understand.